What I'll tell you about today is the title, as Nasreen told you, and this was contributed by two graduate students in my group, Olof Stenström and Carl Deal. They have now graduated, and then a senior colleague, Christopher Modig. And the outline here is like I'm telling this as a story of how we developed this methodology to approach this problem. And then I'll probably introduce or we take a few steps back and look at some very simple-minded NMR explanation of the phenomena just to make sure that everybody is on the same page here. So the problem here is really to try to understand how a protein binds a ligand. And the minimal scheme you could have for protein ligand binding is this four-state scheme here, where this would be on the upper left, which we will call state 1, is the protein in the apo, i.e. ligand-free, unbound state, in a conformation that's not cannot bind the ligand. Well, ligand is here this square magenta shape. So usually the protein has to change its shape in order to bind a ligand, like we have on the lower right-hand side. There's a complementary fit. And then the question is, how does this happen? Does it happen via the so-called induced fit pathway, which I'm sure many of you, or if not most, recognize from classical biochemistry textbooks? This is Koshland's idea of that the ligand binds to the protein and then induces a complementary fit according to this pathway. Or alternatively, what's become known as conformational selection, where the ligand selects a conformation of the protein that is compatible with binding the ligand. And there are more subtle questions involved here besides just determining whether it binds via induced fit or conformational selection. And that is, for example, what are the conformation-specific affinities, ligand affinities? So you might imagine that this conformation has some affinity and this conformation has a different affinity. What are those affinities? Can we determine that? And perhaps even more subtle question is, does the ligand change the energy landscape of the protein conformations? And as I will tell you here in this today, we really believe that, yes, we can determine all of this. So let me introduce the model system that we used. It's a protein we worked on for a long time from many different angles. This is a protein called galactin-3. It belongs to a class of beta-galactoside binding lectins, and it's involved in cell cycle regulation, differentiation, and apoptosis. And for these reasons, it's actually an actively pursued drug target, primarily in disease areas like inflammation and cancer. And what we will be looking at here is just the carbohydrate recognition domain, and this is residues 110 to 235 of galactin. So what you see here on the left-hand side is lactose bound in the very shallow binding pocket, which is really just a shallow cleft on the surface of one side of this two beta sheet clam structure. And in yellow, you see residues that are highly conserved and coordinate the lactose molecule. So lactose is a native-like but minimal part of these galactosides. And usually it's much longer chains, and these chains may continue up here, so there are more binding sites higher up in this. But we will just look at lactose for everything that I will be telling you about here. And as you can see, lactose here has a quite weak affinity. It's like 200 micromolars. And that means that it's actually quite amenable to be studied by NMR exchange. methodology, and we'll see more about this as we go here. So let me tell you the take-home messages before we go further. First of all, we find that galectin does in fact bind the ligand, and via pathway that involves a spontaneous transition from the low energy ground state to high energy state, excuse me, in the absence of any ligand bound to the site. And this alternative conformation actually does have a higher ligand affinity than the ground state does. And that's exactly what you would expect, right, for a process where it binds via this upper right-hand pathway, the conformational selection pathway. But it turns out that galectin binds ligands primarily via this induced fit pathway, except at very, very low ligand concentrations. And the reason for this is that the ligand exerts transition state stabilization of protein conformational change between these two states. So the ligand lowers the free energy barrier in going from this state to that state, but it does it even more so when the ligand is associated to the protein. So this process here has a lower transition barrier than this, and that's the reason why it binds via the induced fit pathway, even though it has all the other characteristics of why it could bind via conformational selection. So you could frame these take-home messages in what actually became the title of our paper that we published last year in PNAS. Ligand-induced protein transition state stabilization switches the binding pathway from conformational selection to induced fit. So now, like in the remainder of the talk, I'll tell you how we did this. And in order to address this question that I introduced, does it bind via induced fit or conformational selection? The name of the game is really to determine all of these individual rate constants in going between the four states. And in order to do that, we measure the exchange rates by NMR. So let me take a brief moment here and look at how we explain conformational exchange measured by NMR. I'm sure many of you have seen this, but just to make sure everybody's following along, I'll go through this in some detail. So suppose we have a protein like the one I've introduced here, who can exist in two states, A and B, and we would have rate constants going KAB or KBA. And it could then, we have two scenarios here, that the protein exists in 10% of conformation A and 90B, or alternatively 50-50. And these are depicted here in the same colors, blue or purple, in these slides. So in the middle panel here, here we assume that there is no exchange. So the rate constants here are both zero. In that case, you would see where you had 50-50, two peaks of equal height, and basically the same natural line width. And whereas if you have skewed populations, then the peak height scales with the relative populations. You would have one high peak and one low peak. And the exchange rate, like I pointed out, is zero. So now if we, so to speak, we turn on the exchange and look at what's happening at very, very fast exchange rates, then you just see an average chemical shift. So it's the population weighted chemical shift of these two states. And its position will then be, of course, be weighted by the population. So the purple lines will be shifted only 10%, so to speak, from its position in the absence of exchange, which is at -100. So that would be basically at 90, -90. And in the case where you have equal populations, you would have it at 0, which is the midpoint of minus 100 and 100. And fast exchange in simulating these spectra here, we've set this to 10,000 per second. So it's clearly much larger than the chemical shift difference between these conformations A and B. Now, if you have slow exchange, so that the exchange rate is slower than the chemical shift difference, and what we've simulated here is an exchange rate of 100 per second, then what you would see is two broad lines of equal height in the case of the 50-50 population. But in the case of a skewed population, you can see here in this representation that the minor state, the one that only is 10%, may actually not be visible at all in the spectrum. But it turns out that the line width of the detected state actually includes information about the presence of this state. So by measuring the line width here, or what we actually do, we measure relaxation rates, and I'll cover that in the next slide. We will get information about the presence and the chemical shift and the population of this invisible state. And from this, fitting all of this data, we will get out the exchange rate, we will get out the chemical shift difference between the two states, and with some additional information we can actually tease out what the chemical shifts are of the two different states, and we get information about the populations. So, you know, I'm a physical chemist and I'm totally ecstatic because this is basically everything you could ever know about a biophysical system, namely the kinetics, the exchange rates, the structure, the chemical shifts, to the extent that you can interpret these. But as I will show in our case, we think we can interpret them quite well. And the population, that is the thermodynamics. So we can basically define the energy landscape of this process quite well, and the structure. And there's, like I said, not much more you ever would want to know about this system. So like I said, we actually don't measure the line width, we measure the relaxation rates. And just to show this, you know, we measure this with spin echoes, so CPMG, Corpuscle-Mabum Gill type experiments. And here I've just shown that if you have on the bottom slide a system, a resonance that does not exchange, the protein does not exchange, so there's only a single resonance that's detected. We have phase on the vertical axis and we have time on the horizontal axis. So this is how the magnetization evolves over time during this pulse sequence. So these are 180-degree pulses, these gray bars here. The slope here is the resonance frequency or the chemical shift, right? So phase, slope, resonance frequency times time, that amounts to the accrued phase. That's the magnetization is swept out in the transverse plane. So then if you don't have exchange, it will look like this, that it evolves, you have a 180-degree pulse, the phase gets inverted, it keeps evolving, inverted again, and then after 4 tau CPMG, you're back at exactly the null zero phase that it had at the beginning, the same phase. And that would amount to having a sharp line with a natural line width that just is the intrinsic relaxation rate. Now, alternatively, you might imagine that there is exchange, and that's what's depicted here. So here we have two random exchange events where the magnetization suddenly jumps and changes its orientation in the transverse plane. And then, of course, you will get imperfect refocusing. Now, this would just be one particular incidence or realization of how one particular spin is experiencing these jump processes between conformations. And that's, of course, not what we measure in NMR. We measure an ensemble measurement. So it's more like this that you have here. You have many. And then you see if you sum these up, this would amount to a broad NMR signal. And the width here, again, would carry information about these exchange events, the rates, the populations, and the chemical shift differences. You see the slopes are different, so that means that the chemical shifts are different between these two, blue and red. So again, we don't measure the line width, we measure the decrease in intensity, as you see here, with time. So obviously, if I would increase the time, we would get even more defocusing and so on. And we can measure that for a fixed time, but instead vary the number of 180-degree pulses. So here we have 3 times as many. And the evolution of the magnetization that's depicted here is exactly the same as up here. The jumps occur at the same positions in all cases, but we have more refocusing events, 3 times as many. And then you see that the width of the signal will change. It will become more narrow, or conversely, the decay of the signal with the refocusing rate will decrease. And that's exactly what we actually measure. So here you see actual data for galectin-3, and you see these decays. You have on the vertical axis is the transverse relaxation rate, which includes the exchange rate that I pointed out previously. And on the horizontal axis, you have the refocusing frequency, the frequency of refocusing events during the CPMG sequence. And here we have 8 different panels. And this is because in our study here, we measured the exchange of the apoprotein, so there's no ligand present at all, and we measure this as 2 fields, 2 different static magnetic field strengths, 800 and 600 megahertz proton frequency. This is N15 relaxation data, and the reason we measure at 2 different fields is that then you can determine the populations and the chemical shift differences a little bit better than if you only have 1. So we did that, and then we measured at 7 different lactose concentrations, increasing concentrations from 4% relative population of bound state to 8, 11, 21, 27, 30, and 75. And we didn't go higher than 75, and I think you can see why. The dispersion here is perfectly almost exactly flat, and that means that there's no more information. It's the change between concentrations that carries the information that we're looking for, that will help us determine all of these exchange rates. And the exchange rate gets faster and faster, and that's in the end here we get into the fast exchange regime, and we cannot refocus the exchange events any longer with the 180-degree pulses. And that is, of course, you may imagine that, for example, the on rates here, for example, this rate, K31 from state 1 to 3, it's an on rate, diffusion limited, and that's scaled by the ligand concentration. So when we add more and more ligand, this process becomes faster and faster, and in the limit, which we hit here already at 75%, it's a perfectly flat dispersion, and there's no purpose going any higher. But it turned out that that's enough. We can actually fit a lot of data with all of this here. So let's focus on how we went about this, and let's focus initially on the data for the apo state. So here in our model, we only have the exchange between states 1 and 2, and you have the forward and backwards rates. And as you can see, we do measure exchange. There's nice dispersions here, and all of these panels are for residue 184, which is a key residue coordinating the ligand, and that's shown here on the next slide, where you see all of the residues that show exchange when in the absence of ligand. And here is 184, which is one of the residues that actually coordinate the ligand sitting here. And you can see there are two different views, orthogonal. They're rotated 90 degrees along the horizontal axis in the plane of the paper. And you see two classes of residues, red, and they are all on the top sheet here where the ligand binding site is. And then we have blue, and they're on the back side of the beta clam structure. And they have actually different exchange rates. So the blue one is a very fast process with a very small population of the higher energy state. Whereas this one here is much slower. It's only 34 per second, and it's a 4% relative population of some alternative high energy state. And since this is in the binding site, this is the one we're interested in. This is decoupled, has nothing to do with this. So from now on, I won't talk about this conformational change here. You might imagine that this is something to do with the loop structure here that's flexing about a little bit, and that's what we see. So we can measure the rate, we measure the population, and we can also extract the chemical shift differences, like I told you before. And we can compare these chemical shift differences. They're plotted here on the vertical axis. So this is what we derive from the CPMG data. And we plot that versus the shift difference that we can measure in spectra, separate spectra, the spectrum of the apostate. We measure the chemical shifts there, and we compare it with the chemical shifts in a sample that's perfectly saturated, so 100% saturated with lactose. So lactose is bound 100% in that sample. And you can see that the correlation between the chemical shift differences between these two states that we measure in the APO is very similar to the chemical shift difference between the APO and the lactose bound states. So that, we conclude, means that this high energy state has chemical shifts that are very, very similar to the lactose bound state. And again, that's exactly what you would expect for conformational selection. So this is a high energy state that's kind of primed to bind the ligand. And it turns out that we think that we have a very high resolution structure of this state, what that is. And it's not determined by NMR, but as it happens, by X-ray crystallography. So we had determined previously, this is an old study from 2012 by my colleague Derek Logan's group, and this is Saravoji was first author. And here we've compared the X-ray structures of the apostate with the lactose bound state. So the protein backbone here sort of diffusely shown, and then you have the coordinating side chains. You see blue and red. The structures are exactly the same. And then in green and red here is the lactose that is bound in the green lactose bound state to the protein. And the pink spheres correspond to water molecules, oxygens in water molecules that are present in the apo structure. So what you can see is that in the apo structure, in the apo crystal structure, there's at least transient population of waters at pretty much exactly the places where the oxygens are in the lactose molecule. So this means that, in solution, once in a while, the protein forms a high energy state, and we think that in that state, water molecules coordinate the side chains in such a manner that it looks just like the lactose bound state. And this is kind of a unique and accidental result that we see that the high energy state in solution actually is the same as this state in the crystal. So that's a little serendipitous result. All right, so we have this exchange, we've determined this now, and again, this was determined in the absence of ligand, but of course, this is the same equilibrium 1-2 as the 1-2 equilibrium when we have all four states present. So even when we start adding ligand, there's nothing changing this equilibrium. We've already determined what that is, and that actually helps very much in when we want to fit all of these rate constants. We've already pinned down what this is, and it's been shown by other people like Mike Rosen's group some time ago that if you can isolate one side of the quadrant here of a four-state exchange, and they did it by mutation, but we can do it here by measuring it in the apostate, then you can actually determine all of these rate constants with some confidence. Right, so next, once we've done this, now we start looking at what do we see when we incrementally here add lactose to the protein, and we repeat and measure the dispersions again. So in that case, we see actually more sites and not the same sites light up. And that reflects that, when the ligand binds in the pocket here, then residues close to where the ligand binds, they will sense the change in magnetic shielding and get a different chemical shift than it had in the apostate. And what I plotted here and what we used for the fitting are 8 residues that show significant relaxation dispersions at all 7 lactose concentrations. There are more residues that show relaxation dispersions at some of the concentrations of lactose, because there's some chemical shift. This is kind of fast exchange, so some peaks change a little bit and so on. So there is some overlap, some shifting around, and we've just to make sure we use the same data, the same residue, so to speak, at each concentration of lactose in the final fitting, we only settled for these 8 residues. And you can see it's kind of similar. In some cases, it's exactly the same that we had in the apostate. So how do we go about fitting this now? Well, it turns out when you stare at these, the matrix of coupled equations, exchange rates, you realize that we had already fitted K12, K21, P1, and P2 from the apostate. So this is fixed. And then you realize that what you need to fit is just the ratio of the the off rate going from state 3 to 1, the ratio of that rate over the going from 4 to 2, the K42. And that's what we call the rho off. That's the ratio of the off rate constants. We also fit the ratio of the closing rate constants. So that would be going from an open state 2 to the closed state 1, K21. And we compare that then with the closing going from state 4 to 3, K43, and that's called a rho close. And then we make the assumptions that the on rates, K13 and K24, are the same. And that's an assumption that one can discuss. It's not necessarily so that it should be that, but we think that in the case of galectin here, it's quite likely that it is the case. Partly because, as you've seen, the conformational change going from apo to lactose bound is quite subtle. And I actually have a slide later where we determine the ground state of apo by NMR, and you'll see how very subtle the change is. So that's one consequence, that there's not a huge conformational change when you go from here to here. And then also we should realize that with NMR, where basically the on rate we're measuring is when the ligand contacts the binding site. So any event like the ligand forms a transient encounter complex, maybe binds on the backside, and then maybe diffuses along the surface of the protein and ends up here, and so on and so forth. All of that, such phenomena should be the same because the structure is quite similar. If the structure had been very different, then maybe the diffusion there wouldn't be the same, and then the on rate wouldn't be the same. And whatever is happening here, all of this is included. So going from the ligand is somewhere out in solution to that it finds its way and then searches the surface of the protein and finally docks. All of that is included in this on rate, and we assume that it's the same for the two pathways. And once you determined these three parameters, I should add also that the total affinity, the total binding affinity is fixed. We keep that fixed, and that's measured by isothermal titration calorimetry under conditions that are virtually identical to NMR. The NMR sample has a factor of 3 higher protein concentration, but otherwise everything is the same. So that's also fixed. And then we only need to fit these three parameters in order to then derive all of the rate constants that we have in this 4-state equilibrium. And the way to do this, or maybe I should say the way we did this, is to simulate the Bloch-McConnell equations, the so-called exchange matrix, for given parameters values. And then we determine the relaxation rate constants from that. We compare it with the experimental data point by data point in the dispersion curve. We evaluate the residuals, the chi-squareds, and then this Bloch-McConnell simulation is then wrapped inside an optimization routine. So we optimize the parameters, and we keep doing this until we find a minimum. And then we do error estimates based on 500 Monte Carlo samples. All of this takes a long time, so fitting data takes several days on a Mac Pro computer, with multiple cores. But it actually converges, and I'll show you here that in order to do that, we first did extensive grid searches on the parameter space, that is the fitted parameters k on, rho off, and rho close, while fitting residue-specific chemical shift differences and the intrinsic relaxation rate. That's the limiting value at high refocusing frequencies of the CPMG train. And you can see here we plot two-dimensional slices through this parameter space. And you see that a low chi-squared is dark blue, a high one is red. And you see that rho close has a pretty well-defined minimum. It's somewhere here, and rho off doesn't like to be very high, so it's also in this area. If we look at the slice rho close to k on, we can see a clear minimum in this regime here, and similar for k on versus rho off. It's a little bit less clear, but it's clearly not the higher values of rho off that's viable. And of course, if you actually reconstruct the full 3-dimensional cube, the minimum gets even better defined. And again, these are the what the different parameters are stand for. Now, one reason why this we can do this and identify the pathways is that the response of these dispersion curves to the refocusing frequencies, so the REX versus refocusing frequency, that response is clearly different depending on which pathway you take. And of course, before we set out to do all this work, we knew that because we had this idea that there should be a signature in the data. So we simulated and knew that this would be the case. But just to illustrate, what I've shown here is what you would expect given the optimal parameters that we fitted, and then say that we only take the conformational selection pathway, then this is what it looks like. So if you go from low ligand concentration, that's yellow, to green, you get an increased quenching of the relaxation dispersions, like so. If you take the induced fit pathway, it's pretty much just the opposite. Then it goes from blue to magenta. So of course, if it's induced fit, if you don't add any ligand, then there shouldn't be any exchange, right? So it's just 0, flat. So this is 12 is the R2 naught value, the intrinsic relaxation rate. And then it increases. And then it kind of flattens out a bit. So it drops down here and becomes higher here at the end. Very different scenarios. And in fact, what we have is this: when you fit the full 4-state model, you get this signature, which is clearly different from these two. So that's why you actually can fit the 4-state model. And I should also point out that the difference here between conformational selection and induced fit had also been realized by, for example, Christian Griesinger and Thomas Weichel's group had a paper, I think, in Nature Communications a couple of years back. But then they only included either this pathway or that pathway, whereas we now show you can fit both pathways, all 4 states like this. And of course, you have to be a little bit fortunate, I think, with parameters in your system for this to work, you know. Okay, so here are the results, all the fitted parameters. And you can see that we have pretty good precision in the parameters. They're like maybe 4 to 20% error for most of them, in some cases even lower. And from this data, we can actually extract a wealth of information about the system, some of which is quite surprising. But let's start just by looking at these two rates here, the off rates from the two sites. So as you might imagine, from state 3, that's the initial ligand-bound state for the induced fit pathway, where the protein is kind of docked onto a non-compatible conformation of the protein. That off rate is quite high, whereas it's much, much lower when it's the off rate from the actual final state of the complex. And this means that the relative affinity of state 2 over state 1 is 270. So it's 270 times higher affinity for the conformational selection pathway, so to speak, or for state 2, that's the first step in that pathway. If you calculate the total affinity across the pathways, it's still a factor of 20, 19, in fact, more likely to bind via the conformational selection. But this is just the affinities, right? This doesn't actually tells us which pathway is dominating. And I should say, of course, that both of these values is exactly what we expect for conformational selection. Then this is what you expect. You have higher affinity for the state 2 than state 1. But in order to determine something about the pathways, you have to look at the flux. So that's kind of a chemical engineering language. It's the mass transfer through each of the two pathways. And here are the equations. This is the flux through the conformational selection pathway. This is the flux from the induced fit pathways. And this is given by these expressions. I should point out that the first term here, that's the inverse of the waiting time. Oh, sorry, this is the waiting time in state 2 before it leaves to go to state, in state 1, in waiting before it leaves to state 2. And this is the waiting time in state 2 before it goes to state 4. So the flux is just the inverse of the sum of the waiting times at the intermediate states before you end up in the final state. And likewise here for induced fit, this is the waiting time in state 1 before you go to state 3, and this is the waiting time in state 3 before you go to state 4, and then it's the inverse of that is the flux. And so this we can calculate again from the parameters values that we fitted, and the result is shown here in these graphs. So here you see the full curve and conformational selection in blue, induced fit in red. And you can see, for example, our highest lactose concentration was 75% saturated. It's here, and it's, of course, the binding pathway is occurring almost exclusively via the induced fit pathway. And if we expand up this region here at very low ligand concentration, low degrees of saturation, that's shown here on the right-hand side, and you see that there's a crossover point at a very, very low ligand concentration or degrees of saturation. So, at very, very low concentrations of ligand, there is actually the conformational selection pathway dominates, but as soon as you add just a little ligand, it switches over, and then they both have a significant contribution until you reach, where we stopped at 75, then it's just one, the induced fit pathway that dominates. So what does this mean? And why is this the case? And that's what we can see here. If we compare the rate constants going from state 1 to 2 with the rate constant going from state 3 to 4, so in both cases we're going from the binding incompatible conformation of the protein to the binding compatible conformation, and you see that it's almost 1,000 times faster when the ligand is present, when the ligand is docked onto the protein. And this is the same thing as saying that the ligand induces transition state stabilization. So this is just a representation without any claims of making this the various levels here accurate in terms of what we measure. But you have on the induced fit pathway, the blue one here, the barrier height going from state 3 to 4 is much lower than when you go in a conformational selection where the ligand is not present. Here's just the empty protein. Higher barrier when the ligand is present, it's a lower barrier. And I'm sure it occurs to you that this is exactly the same phenomenon as how an enzyme works. Enzyme lowers the reaction barrier for some reaction. Say that you have something where the ligand changes conformation and the enzyme catalyzes that, like it could be cis-trans isomerization of peptides like cyclophilin does, for example. And so it's a very well-known concept. It's just that this is the opposite side of the coin, so to speak, where this is the ligand giving a transition state stabilization of the protein changing its conformation. So, from a physical chemistry perspective, there's nothing surprising here, but it's in biochemical literature, in biology, I don't think it's very well, it's not a common idea that this is the case. So that's the full story, and I think I'm pretty much on time here, maybe a little bit over time. Again, thanks to the guys who did the work, Olof, Carl, and Christopher, and to some nice people for funding. And then I should stop here and thank you all for attending and listening. And I'm happy to take questions.
Thank you, Professor, for the fantastic talk. And yeah, let's open the floor for questions. I see that we already have questions. Yeah, so we have a question here. Did you observe similar trend of the CPMG data in the same region? Do you think any allosteric effect can contribute to the exchange rate?
You could absolutely have allosteric effects contributing to the exchange rate. But here, and we've seen that in other systems, but here's there's not really any allostery to speak about, right? Because we only have the one ligand. So from that perspective, there's nothing in our data to indicate allostery. One might be able to, it's known that galectin, there may be modulators of binding and so on. So for example, galectin has a long intrinsically disordered region, and then you have a folded N-terminal domain, and that N-terminal domain can actually come back and dock onto the C-terminal domain, and so some effects like that, if you were to look at it intact protein, that may give rise to allosteric effects. But in our case, there's nothing like that.
Okay. Well, I have a question. I'm not sure, maybe it is similar, but so maybe I missed the explanation, but why the high energy conformation was corresponding to the apo crystal? Like, what's the explanation you think?
No, it is because we think that for some reason, we think the crystal contacts in the crystal selects for the high energy conformation, and that it's also stabilized by the waters that are then docking onto the hydroxyl binding sites in the protein. So, you know, the high resolution structure is a cryo structure, so it's done under liquid nitrogen conditions, and that of course stabilizes even more. But we also collected data at room temperature, and you still see signs of electron density from the oxygens in waters in the similar positions. And we looked at it actually, the water coordination also by molecular dynamic simulations, and then you see increased transient population of waters sitting in these sites, the same sites basically. We also measure the water exchange dynamics by another type of relaxation dispersion technique, measuring the waters themselves. And from there we can say that all of these waters in the binding site, they exchange with the bulk water on a time scale of nanoseconds, so very, very rapidly. But it means that at any given point, there is an enhanced population of waters near these hydroxyl binding sites. And then once in a while, you know, there will be waters presumably in all of the sites at the same time, but that's a very rare event. So there's a very low likelihood that it happens. So you have a very low population. You only have 4% of that state present. So that's how we think about it. Yeah.
Okay, interesting. Cool. Yeah, we see more questions. So we have a question here. Bonjour, I'm confused with how the parameters are fitted. Could you please go through all the assumptions again and talk more in detail about the fitting parameters?
Yeah, okay, I'll do that. I'll
for
back
Mikael.
up. I think I'll need to do that. Back up again. Let's see if we can do that. There we go. I think it's this slide. Yeah, again, when we measured on APO, and that means we can fit these parameters, they're already known. And then we make the assumption that the on rate for each of the two pathways is the same. And again, I argued the case why we think that is the case. We've actually tried fitting with separate on rates, but as you might imagine, the precision and the errors become lower in that way when you have two free parameters instead of the one for this. And then we have the ratios of the rate going off rates. In this pathway, K31 over K42. So that's the off rate on the conformational selection side. And then you have the closing rate. That's the rate going from the open state 2 to 1 or the open state 4 to 3. And it's really only these we fit. And then you have the total affinity for all of these, for the total binding of going from this to these states here. So it is actually the case, if you look at all of these, the populations in the end, you actually have a subpopulation in the fully saturated state of state 3. So that's something we actually took into account when we used this KD and partitioned it out to the different states. And I think I actually have a slide further forward. Let's see if I can jump to there where we can, all the equations are expressed. So here's basically how you relate then the KD to the microscopic states. Yeah, it's a little bit hairy, but in the end you end up here on this right-hand side here. And likewise the K-offs for the different sites. For example, from site 4, you can relate to these different sites and so on and so forth. So this actually means that you only have those 3 parameters to fit. Let me go back. Where were we? Here. And from this, we can then determine all of these different rate constants. And from there, like I showed you, we can extract other parameters. I don't know if that helped, or feel free to just follow up with more questions if you want to know more.
Yes, actually, we have a follow-up from the first question. So, allosteric, they meant by like the binding of a cofactor, like calcium or magnesium, if they are present.
Yeah, no, but there's, as far as we know, there's no magnesium or calcium present in the sample. But it's very clear that allostery can affect these types of dynamics. And again, we've seen that in other systems. We published a paper in Science Advances on the glucocorticoid receptor, where you see the effect of allostery on the exchange rates in the protein. But many other people have done similar studies as well. So it's clear that allostery can have these dynamic effects.
Okay, cool. Hopefully this answered the question. We have another question. Is it correct to assume that the K-on is the same for both free states, only in the particular case of galectin 3, where the conformation of the free and bound states are the same? Yeah, sorry. Were they the same or no?
Yeah, no. So I think in many cases, I think you could say like this. In general, you could use that assumption, except in cases where you know that there's a big conformational change. Suppose there's like a protein, one of these states would be like this, and the other one would be a big, huge domain opening. Then it's clear that a ligand coming in to, I'll use my fist instead, like this, a ligand coming in and transiently forming a transient encounter complex here, and then maybe diffusing until it finds the binding site. That diffusion will be quite different than if you have a shape like this, and it docks here, and then it diffuses into maybe a binding site between my hands, So, but as long as the conformation of all the different states are quite similar, which is the case for galectin here, then I think it's a pretty safe assumption. Because the diffusion through solution, the actual diffusion limited on rate until it finds the protein, that has to be the same.
Okay.
Another caveat would be, for example, if you have if there's a charge interaction, Coulombic interaction, so the ligand is charged, the protein is charged. That's not the case for galectin. But if you have a case like that, and the protein, a conformational change in the protein changes the charge distribution, then that could actually even also change the diffusion limited on rate. So the diffusion can be steered by electrostatic interactions to some extent. But again, that problem we don't have in this system.
Okay, cool. So we have, could you explain why K31 is equal to K24? Exchange, yeah.
K31 is not equal to K24.
Yeah.
K13 is equal to K24. That is this assumption that the on rate is the same that we just went through. Yeah, so
Okay.
if there was a misunderstanding that it's K31 is not equal to K24, but these two are equal. K13 and K24. I'm sorry, I may have misspoken, but these two are equal, the on rates.
Okay, another question. You assume that K on is the same for both processes with different conformations because the conformational differences are small?
Yeah, that's one reason why we think it's a good assumption. I think
Okay.
we would probably have started with that assumption even if we didn't know anything about what the conformations may look like, and then see if that would fit the data. You might imagine that if the K on rates would be quite different, then maybe you wouldn't be able to fit the data either. But yes, it is the fact that the conformational changes are quite small. Maybe I should actually show that. So I mean, we've determined.
Yeah, I mean, there's a follow-up. So then he said, if so, why does a small conformational change need to overcome a large energy barrier? So 34 over second seems like a relatively slow process. So it should have a high energy barrier.
Yes, that's right. Yeah,
Okay.
it does. So here, let me start with this. This is the ground state APO structure in red, an ensemble of structures determined from only backbone RDCs. So there's very little, and also actually this the side chain here, tryptophan side chain we had data for. And in green and blue, again, green is like before is the backbone trace of the lactose bound state. Blue is the backbone trace of the apo state. And you see again, like I pointed out before, the blue and green superimpose almost perfectly here. Whereas the red, the ground state, apo in solution, deviates a little bit. And again, binding site is here where my show with the laser pointer. So if you see that now that the changes are basically just out in some loop regions. So if we look at the protein from the side, here's the binding site. I don't know if you can see my hand. Then the ground state is a little bit more open, like so. And then when it binds, it comes in a little bit. And moreover, we think that this tryptophan is actually moving a little bit and docking onto the protein. So there may be more of a change here that we didn't pick up because we only had, I think, one probe, RDC probe, in this region here. The indole NH, I think, was the only one we had. So that's that. What was the question now? There was another question. Oh yeah, the energy barriers. So I don't know if I should go back just to illustrate that again. This slide. So could you repeat the question relating to the energy barriers?
Yes. So the energy, why does a small, yeah, why does a small conformational change need to overcome large energy barrier? And then the value 34 seems like a relatively slow process. So it should have a high energy barrier.
Yeah, so the K12, which is the slow process, has the higher energy barrier, right? As you would expect. And then the K3 to 4 which is much faster, has the lower energy barrier in this scheme. Again, the end point here, of course, is there must be lower energy when the protein ligand is bound than when it's not. So that's just why these levels are different. But the difference here is the largest, right? And you could then, so you're asking why is this rate so slow when the conformational change is so limited? Well, from what I showed you of the ground state, that's just a backbone trace. And you should think about all the different side chains that coordinate the ligand. In the high energy state here, they are then pretty much aligned and in the same way as when it's bound to the ligand. And the waters are presumably organized in a way like that. And getting that concerted conformational change is a very rare event. So that's why it's a slow event, conversely, and the barrier is high. So that's how I think about it. So it's a little bit misleading if you only look at the backbone trace, which is quite similar, whereas there's presumably some change of the side chains at the same time.
Okay, hope this answered the question. We have another question. What software was used for fitting the data for the K exchange or exchange rates?
Yeah, it was home-written MATLAB scripts. And the code, I think the code is available on GitHub. It's not the cleanest, but we decided to just put it out there just the same. Yeah.
Okay, cool. Nice, thanks. What's the expected physiological sugar concentration? I mean, I guess it's the milk sugar, yeah.
Yeah, so I mean, the natural ligand is actually not low molecular weight disaccharides or so. It's more carbohydrate chains in the extracellular matrix and so on. And in the extracellular matrix, depending on where you are, and galectin actually forms these raft-like structures on the outer membrane. So the local concentration of these can be quite high. And but intracellularly, these concentrations are presumably quite low. Okay, galectin is actually a funny protein because it exists on the surface of cells. It exists in the cytosol and it exists in the nucleus. So it's one protein that migrates depending on cell cycle state and so on between different environments.
Okay. Have you done a square calculation using T1, T2, NOE experiments in increasing ligand concentration?
No, we haven't, but we have the data for the lactose bound, fully saturated, and the apostate. And we fully expect that any intermediate concentration is just a population weighted average between those. So I think it would be not so meaningful, perhaps. We published a lot of data on this. That's a whole separate story. We tried to use order parameters as a proxy for looking at conformational entropy. And then once we know the entropy, we can then start dissecting total entropy into conformational entropy and solvation entropy. So we published a number of papers on studies like that for both lactose, but also drug lead compounds and fragments and so on.
Okay, nice. And hopefully, no, just kidding. Last question, why did you assume that state 1 and 3 have the same energy?
That's just arbitrary. Yeah, sorry. Just to put like I tried to say that the actual scales here and where they're positioned, this is just a depiction. So, but just to put them as a reference point, state 1 and 3 in the same position, then you can see the differences in the barrier heights a little bit better. Yeah, sorry if that wasn't very clear, but it's clear that they don't have the same free energy. They shouldn't.
Okay, nice. Actually, last question from Julie. Would this work for any KD value, or is there a range where this approach would perform better?
Now, I think you need to have a system where you will get relaxation dispersion due to the exchange. If it's a very slow exchange, you could maybe measure it, with longitudinal exchange, spectroscopy and so on, and via that fit rates. But I think in many respects, we were quite lucky. So, NMR is very good at screening for binding when you have fast exchange or intermediate exchange. Like this is, depending on the chemical shift difference, some residues here are more slow than others and so on. So we're in the intermediate to slow regime in this case.
Okay. Actually, we have a last question right now, or maybe two. CPMG of the nitrogen group or side chain methyl group, it seems that ligand binding does not cause backbone deviation severely.
No, yeah.
Okay.
Sorry, I don't get it. What's the question?
Oh, like CPMG of nitrogen group or side chain group. Maybe did you do those? Or commenting about the nitrogen or
So
the
we
side chain?
only have nitrogen in this case, and
Okay.
well, you saw there's a handful of residues that get significant chemical shift difference between free and bound, and hence significant dispersions. So it's quite likely that if you were to measure with other side chain probes, you would see more data, have more probes of the process.
Okay, we have another question. In the flux graph, you explained that the conformational selection and the induced fit at very low concentration showed intersection. In what cases, if possible, that only one pathway dominates?
That would be the case. So, for example, if you were to say that for some reason these barriers would be the same, then the conformational selection would dominate, I think. And
Okay.
yeah, so that's just one case, but one can play around with these rates and see various scenarios where you would get dominant pathway over the other. But in principle, if you have 4 states, you pretty much always have a combination. Then the question is just how much does one of the pathways dominate?